So how do you factor in the environmental condition? Because in the beef industry, we've got cattle in the swamps of Florida and in the coldest climates in Sydney, Montana. How do you factor the environment that these cattle will grow up in when you're looking at the DNA level?
Well, that's a really good question. So this is, in the equation, this is the so-called G by E, right? The genetics by environment interaction. And we have the ability to get to some of that. Some of that's driven by, the fact that in certain environments, certain breeds predominate. So take for example, like South Texas versus, Sydney, Montana. Well, in South Texas, we use Indicus cross animals, with the Brahman, Brayford, et cetera, et cetera. And what we know is there's quite a lot of difference in the genome sequence of the Bos Indicus, that is the Nellore, Brahman, Brangus, et cetera, from the Bos Taurus animal. So we can use some of those differences to say, okay, those animals that are more heat tolerant, they have these kinds of genome sequences, and animals that are, the more straight bred Bos Taurus have something somewhat different. So we can use breed to sort of get to the G by E, but we can also begin to investigate that, in and of itself, even.
Let me clarify that. Because there are Angus cattle in South Texas, there are Angus cattle in Sydney, Montana. Our genetic evaluation has always been based within a contemporary group, and you can't compare Santa Gertrudis or Beefmaster against an Angus. But are you telling me that we can look at the DNA of the Bos Indicus cattle, the more heat-tolerant cattle, and it'll tell us something about the Angus cattle and their propensity for heat tolerance?
Well, I wasn't really sort of getting to that point, Trent. What I was saying is that when we look at breeds that do well. So, for example, the Santa Gertrudis example is a sort of a breed that's been built for those sort of more tropical kinds of conditions as opposed to more temperate conditions. And what we see is that there are elements of the Bos Indicus animals that probably contribute to their suitability for a certain environment. So we know that they're suitable for a certain environment, and the differences in their genome is what makes them suitable for their environment. But you touched on the issue, which is, well, what if I have an Angus animal in this environment and an Angus animal in this environment? Do I find, a different response with a similar kind of a genetic background? And that's sort of this G by E interaction. And the answer is yes, we are, starting to investigate some of those kinds of things. An example I might give you it would be, for example, response to a treatment. So a treatment would be an environmental impact. Okay, so let's say, for example, I have an implant. You talked about implants previously. So let's say I have an implant, and I might want to be able to say, well, which animals respond to that implant and which do not respond? Which animals have the genetics to respond and which animals do not have the genetics to respond? So that would be, which Angus cows do well in hot conditions, which Angus cows do not do well in hot conditions. So yes, the answer is that there are lots of studies going on that are telling us, what is it about certain individuals within a breed that make them adaptable to a certain environment. Example that some of your readers or listeners would be interested in would be, say, for example,
I got the perfect one, because we're in St. Joseph, Missouri. Which genetic lines of Angus cattle, we're talking about Angus today, are fescue tolerant?
That was the point I was going to make, and that is that there actually has been work underway to look at tolerance to fescue toxicity. People have been researching that, and we're now starting to identify some of the genes that we think contribute to susceptibility to fescue toxicity. So, once we've completed a little bit more of this work, the prospects are good that we might be able to actually identify animals that have a sort of a genetic tolerance for fescue toxicity. And ironically, some of the fastest growing and most productive, fescue is the stuff that is that plants that are most susceptible to the infection with the fungus that gives you the endophyte. So, yeah, the problem is the difficulty is some of your most productive pastures can be some of your worst affected. But as I said earlier, there has been some work ongoing, and there are some exciting developments that would actually direct us, because ranchers will tell you some animals are affected more severely than others. There's obviously a range of tolerance to the problems with endophytes. So I think the prospects are very good. I wouldn't say that we're there yet, but there's some very promising leads in this area that should give ranchers, I think, a lot of confidence that ultimately we may be able to have a lot of grazing land that can be used much more effectively.
And everyone has a vested interest in that, whether you ranch and graze fescue or not, because once you determine how you can identify the genetics which are fescue tolerant, the next step is we determine the genetics that are resistant to E. coli 0157H7, because I believe, and we have a little bit of data that says that there are certain genetic lines within the beef industry which are resistant to such things as E. coli 0157H7. So once you open the door, the floodgate will follow. And I think, Stewart, that it's important, and you come from a completely different aspect than I do, because you're out explaining these things to the benefit of the rancher. Everything that I hear you talk about, I want to position it as, here's what the rancher's doing and how it benefits you, soccer mom in Kansas City, whether it be through a greener environment, whether it be through less feed utilization, less water contamination, or simply a more safe food supply.
Yeah, and I think that, so if you go back to some of the things, we've talked about things like efficiency and, carbon footprints and things like that, and how we can reduce, that by using more efficient production, how we can make better use of grasslands, including those that are infected with endophyte. Food safety issues, susceptibility to 0157, all of these things. I think, obviously, Trent, these are things we're working on now. I wouldn't want your listeners to think we've got the problem solved by any stretch of the imagination. But the great thing is if we capture the right phenotypes and we save, samples, and we can look at the DNA, we can now start to really be able to much more effectively identify the favorable animals that will produce the product that meets the consumer demand. I talked earlier about, application of technology for the benefit of the cattle producer and application of the technology that produces a product that's safe, wholesome, and desired by the consumer. And there are really two goals. One is to help the rancher in what he does every day, and the other side of it is to produce a product that the consumer desires, finds, great to eat, and is very safe.
All right, Stewart, the Indians have a powwow in a circle because they believe life is a cycle. There's no ending, no beginning. We started talking about me as a mule, so we're going to end talking about the benefit of a mule. The first genome sequencing that I knew of was Idaho Jim. Do you know the story of Idaho Jim?
No.
At the University of Idaho, they cloned a racing mule and have been studying his genetics and his genes because horses and the equine species have a significantly lower mortality rate when exposed to cancer. So through cloning and looking at the genes of this racing mule that they raced for publicity, they're actually using gene sequencing to find a way to beat cancer for human beings. And that's exciting stuff.
Well, and we began our broadcast when I talked a little bit about sequence and the similarities, the homology between the species. And Trent, I would tell you that has been one of the great, so if you will, byproducts of this, and that is that animals have proven to be wonderful models for some of the human diseases, and the discoveries that we're making in the animal genome, whether it's the pig, the cow, the dog, all of those things are contributing to our knowledge of these disease processes and are contributing to advances in human medicine. And I have many colleagues who work diligently in this area, and really, I think there have been some tremendous breakthroughs. So, I think there's a lot of good that can come from this, not only for the rancher, but for the consumer, and also for the contribution to medicine.